All right. Thank you, everyone, for allowing me to moderate this session. I'm Rashad Aita. I'm a master's thesis student, the experimental surgery program. And as I said, I'll be moderating today's student presentation. I hope everyone's having a nice afternoon and is ready to meet two wonderful speakers today, Kayla Bourque, a PhD senior candidate, and Dr. Terry Hébert, who's also my lecturer for family medicine class. So allow me to first introduce the student presenter, Kayla Bourque. She is a PhD candidate, as I said, who is pioneering the use of stem cells in Terry's lab. One of the main aspects of her thesis focuses on comparing the signaling events in primary versus induced pluripotent stem cells derived cardiomyocytes. She is also interested in understanding how signaling modalities change as a consequence of disease, and is currently assessing this in iPSC-CMs derived from patients diagnosed from idiopathic dilated cardiomyopathy. She's presented something similar in one of the MRM talks previously, but in today's session, she will present some data comparing signaling in rat versus iPSC derived cardiomyocytes. It's essentially a head-to-head comparison between the two model systems. And after the presentation, I'd like to ask everyone to feel free to ask any questions via chat or unmute if you'd like to. We'll have a nice Q&A session right after. And in the meantime, Kayla, would you like to put up your, to share your screen?
Yes, let me do so.
So her presentation is entitled Single Cell Profiling of Signaling Networks in Primary Neonatal and iPSC-Derived Cardiomyocytes. Kayla, I think without further ado, the floor is yours.
Great, thank you so much for the kind introduction. So as mentioned, apologies if you guys attended the MRM talk, because this is a very similar presentation, but I hope you enjoy it nonetheless. So essentially, at the heart of our lab, essentially at the core of it, so we're interested in studying how the functioning of different GPCRs, and I'm interested in studying how GPCRs function in the heart. And essentially, when you activate these receptors, it results in it regulates heart rate, contractility, growth, and vascular tone. And essentially, and I guess unfortunately, in some pathological situations, essentially, these signaling pathways get dysregulated, and it could result in disease through different mechanisms, such as apoptosis, hypertrophy. And this essentially progresses into the development of cardiomyopathies as well as heart failure. And a lot of what we know about GPCRs and how they function has been studied using immortalized cell lines, such as the 293 cell. Primary animal cardiomyocytes have also been used, but now essentially we have the ability to use these novel cell systems, so those derived from stem cells. Essentially, since they're from humans, it increases the physiological relevance of our assays being performed. So in this talk, as introduced, I'll be examining cellular signaling using a more established model, so essentially primary neonatal cardiomyocytes, and comparing them to this newer model, so human iPSC cardiomyocytes. So in the lab, we've established two model systems, so either from rat or from human sources. Both of these models stain positive for the sarcomeric marker alpha-actinin 2. So besides the species differences, there are other differences between these two models. So for instance, rat neonatal cardiomyocytes are essentially in an in vivo context about 5 days prior to our assays. This is because these cells typically only last in culture for about 7 days. And that's in comparison, I guess, to iPSC-derived cardiomyocytes, where essentially after day 7 to 10, you can see here, they spontaneously beat in culture, which is a nice indication that essentially the differentiation protocol worked. And after day 28, and I guess until 3 to 6 months thereafter, we could use these cells to perform signaling assays. So these cells are signaling competent. So we performed an RNA-seq experiment, and we looked at here basal GPCR expression, and we can see that They express relevant cardiac GPCRs. And there are some differences. So for instance, in the rat neonatal cardiomyocytes, they express higher amount of alpha-1 adrenergic receptor, whereas the iPSCs cardiomyocytes express more of the AT1R, one subtype of the endothelin, and the beta-2 adrenergic receptor. Both cell types express relevant G proteins. And if we look at the expression level of, for instance, the beta-2 adrenergic receptor over time, we can see that its expression is relatively maintained, suggesting that if we perform experiments at day 28 or let's say 48, it shouldn't lead to I guess, measurable differences. However, we do see some differences here for the AT1R. It seems that the expression might decrease over time. However, these cells are still incompetent and they express the relevant receptors and effectors. So to give a little glimpse at our pipeline, essentially, now that we have our cardiomyocyte cultures, where essentially, in order to be able to study cellular signaling, we transduce them using viruses with our FRET sensors of interest. So these are biosensors that are essentially able to monitor kinase activity, for instance. So if a kinase of interest is activated, it can phosphorylate the substrate domain of the biosensor, resulting in a conformational change in the sensor, increasing the FRET being captured, using here a high content imaging system. And then after, we essentially feed in these images into our single cell analysis pipeline that's been developed by a previous PhD student in our lab. And it allows us to parse out how different cells behave within our overall culture. So looking at either non-responders, cells that respond low, intermediate, or high. And then we plot this as a heat map to visually inspect our cells. And this essentially is in contrast to a bulk analysis, where essentially you're just averaging out thousands of cells into a single point over time. So this is essentially what our heat maps look like. So this is looking at cytosolic ERK activity measured using our biosensor called EKAR-NES. So here on the y-axis, these are all individual single cells. On the x-axis, it's time. So green here depicts an increase in FRET, so an increase in kinase activity, whereas purple is a decrease. So when looking at our human iPSC cardiomyocytes, there's not drastic, I guess, activity being measured, especially compared to vehicle. So the patterns here of the heat maps are quite similar, so that's suggesting that we were not really capturing large amounts of activity. In rat neonatal cardiomyocytes, we do see that in response to SI and SII, which are two beta-arrestin bias compounds, we do see about perhaps 20 or 30% of the population that are resulting in increase in ERK activity. So based on this data, there are some subtle differences that can be seen. When looking at agonists that target essentially the alpha and beta adrenergic systems, so isoproterenol, we can see here a large population that actually leads to a decrease in FRET being measured. So this, in a sense, would suggest that perhaps here this drug is behaving as an inverse agonist. Endothelin here leads to a robust activation, where you can see perhaps half of the population leads to ERK activity within this overall population. When looking at rat neonatal cardiomyocytes, now we see populations responding to norepinephrine and epinephrine, and this might be due to the fact that essentially these cells express more of the alpha-1 adrenergic receptor. Also seen here the fact that they respond to phenylephrine, which targets this receptor system, and again we see a nice robust response to endothelin. So now if we switch looking at cytosolic PK activity using another biosensor, we see here three nice populations being, or nice clusters being seen. So for instance, in response to isoproterenol, we can see there's a population here that are low responders, then we have intermediate responders, and then robust responders, and these responses are essentially sustained over time. We see a similar pattern in rat neonatal cardiomyocytes. However, phenylephrine here does not seem to produce as much of a PK response. This is in contrast to data collected from another PhD student in our lab who showed that phenylephrine leads to a nuclear PK activity. So I'm currently assessing that as well, and I do have some preliminary data that does show that we do get nuclear signaling. So this could be, we could see some compartmentalized signaling within these cells. Now switching to drugs that target the AT1R. So in the rat system here, we can see essentially what is expected. We don't really necessarily see PK activity, considering that the AT1R is essentially coupled to GQ. Interestingly, in the iPSC model, we do see perhaps small populations of cells that are responding to a small degree. So this is something that is interesting and that we might follow through to see if these responses are specific to the AT1R or if we're getting off-target effects. So right now, we're also, I guess, complementing our signaling assays with hypertrophy. So essentially, in rat neonatal cardiomyocytes, we have shown that stimulating these cells with endothelin, so at 24-hour time point, essentially, we can see an increase in cell size, shown here using microscopy, but also quantified using these bar graphs. And this is true for endothelin, phenylephrine, and other drugs here, so isoproterenol, for instance. When using our human iPSC-derived cardiomyocytes, using a microscopy approach, we have not necessarily been able to show this increase in cell size, but this might be due to the fact that at baseline, we have cells with different sizes, so we have a larger spectrum, and this may essentially drown out any individual increase in size. So using a single cell approach for hypertrophy might be the way to move forward. However, when looking at gene expression, we do see that endothelin does lead to an increase in ANP and BNP, which are two hypertrophic markers. So in summary, I've shown so far that there are subtle differences observed in the cellular signaling signatures in both stem cell and primary derived cardiomyocytes. And an ongoing question in the lab is that we want to assess essentially how the signaling signatures might change when we mature our iPSC cardiomyocytes, considering that right now they're more, or they're not as adult-like as we would hope. And also, we're also trying to complement our current data sets with signaling data derived from rodent stem cells. So essentially, we want to see if the rodent iPSCs are going to be closer to essentially the human model or the primary model, considering that there are differences in the way that they're actually cultured to begin with. And essentially, future directions of our project, now that our signaling pipeline is essentially well established in the lab, we were taking cells derived from patients who have been diagnosed with dilated cardiomyopathy, and we're going to be applying our same pipeline here, so transducing them with our biosensors of interest. And we're essentially hoping that we'll be able to find clusters, so some cells behaving differently, and that essentially these clusters could help us develop rational treatment options for these patients based on our bench work. And of course, hopefully this will lead to improved clinical interventions. So that's it. Thank you for listening to my talk. And I would like to thank my supervisor for his continued support throughout the years, and as well as everybody else in my lab who has helped me, especially Jace, with the whole single cell analysis pipeline.
Thank you, Kayla. That was a very interesting presentation. Well, I thought all of this was interesting when I first read it. Now I know it's way more interesting.
Thank you. Should I stop sharing my slides or?
Yeah, you can. So now that the presentation's done, unfortunately, we can go on to the Q&A session. I think Dr. Mwale has a question, yeah?
Yes, Kayla, that is wonderful. Really great work. I enjoyed it. So the human iPSCs that you showed, and you are looking at the EK signaling. So are those from male or female? And because one of the conclusions you gave was that they are you observe signaling signatures. So would they be different from male and female?
So the stem cells that I showed in this presentation are from male. So they're male that are, I believe he's in his 30s or 40s. But I do have other data, essentially from male and female patients in this case, that do show some differences. So that is a question that we do want to address to see if we're able to see differences between male and female control, and of course patients as well, because the use of iPSC models allows us to actually ask this question. Whereas when using primary cells, for instance, we're kind of just putting all the pups together, and we're not really considering sex here as an endpoint.
Okay, so when you compare to rodent iPSCs, what would they be close to? Would they be, what sort of differences do you expect? Would they be close to male or female? What?
Well, in the litter that essentially we use, these are like 1 day old pups. So there's a mixture of male and female. So that is something like using this system, we're not necessarily going to be able to address that due to the confound here that we're not essentially controlling sex in our primary cultures. But we'll be able to do so when we're doing using our patient models.
Thank you very much. Thank you. That was wonderful.
I think we have a question in the chat. Okay. What was the primary cause of the cardiomyopathy?
So the patients that we have access to, they have dilated cardiomyopathy. So those are the patients that we're going to be using to assess different signaling and morphological characteristics in attempt to better understand the mechanism of their disease. The data shown today was just control. It was not from stem cells derived from patient cells. I believe Terry might show some of that.
I have a quick question for you, if you don't mind. Well, you mentioned like the versatility of iPSC-CMs in this context. I wanted to ask you, how do you plan on maturing these cells to reaching to the desired passage or desired function?
Yeah, so essentially, I'm not addressing it myself in the lab, but we have another graduate student who will be doing this. So essentially, we're going to be trying first using different media formulations. So essentially, I guess neonatal myocytes use more glucose as a primary source for metabolism, whereas adult use more fatty acids. So we're going to be trying to supplement our cultures with fatty acids and other hormones that have been essentially published in the literature and have been shown essentially to promote a more adult-like state. So we're hoping that this is going to work in our hands. So we've tried a couple of times. We have seen that the fatty acids do lead to some level of cell death. So now we're in the process of kind of titrating and optimizing the actual concentration of fatty acids that are optimal for our cells, essentially.
Thanks. Any more questions? Anybody would like to unmute if they'd like to, or ask in the chat? I think we have time for 1 or 2 more questions.
Hi, it's Jean Trevenkov. I'm not a cardiologist or a cardiac surgeon, but I got interested in cardiomyopathies because my understanding is a lot of them are immunologically induced as a result of viral infections and targets of the cardiomyocytes. And that was my question as to what exactly was the cause of the cardiomyopathy. I mean, I understand they're eventually dilated, but what's the primary cause in these patients? Was it related to an autoimmune disease or a viral infection? And
so
therefore,
it's diverse. Yeah, sorry.
It's diverse, I understand, but particularly those patients you presented. Do you know the history?
Yeah, so we do have the data provided from our cardiologist, who's also working with us in the project. So some are from viral sources, others genetic, some are idiopathic, but that's probably just due to the fact of there's not enough genetic screening being done. There's postpartum causes. There's some that are alcohol-induced. So we have, I guess, a large spectrum of different causes.
Because ultimately, the mechanisms of cardiac destruction may be similar. And my question is, do you study this in conjunction with the immune system? Or is this totally independent? Because if you throw in stem cells that express the same antigens, you're not going to cure this disease.
No, that's a good point. So right now we're just working with cardiomyocytes and trying to better understand what's going on in these cells. But I know that eventually I guess in future directions of the project, we want to use co-cultures and also organoid models to better understand how this is going to work. Right now, we don't necessarily have any plans to look at the immune system per se, but this could essentially develop over time. But it's a very good point that you bring up to us.
Thank you.
I will show our patient population.
Okay, I think we don't have any more questions. So thank you, Kayla. It was a really nice presentation. And
thank you.
No problem. So I think we can move on to Dr. Terry's presentation.
Yeah, so I will, so thank you very much, Dr. Hébert, for coming to give us this wonderful talk and for such a short notice. And Kayla, thank you very much for that wonderful talk. And Rashad, nice, wonderful moderation. So Dr. Terry Hébert is a professor and graduate program director for the Department of Pharmacology and Therapeutics at McGill. He is also the director of the McGill Regenerative Medicine Network and the Canadian Pacific Chair in Biotechnology. So he has a lot of interests, and they lie mainly in understanding the biology of GPCRs and G proteins, with an emphasis on oligomerization, signaling complex assembly, and localization to the nucleus. So he has a lot of focus, and one of his current research focus is looking at ontogeny, formation, and trafficking of GPCR signaling complexes to understand the architecture, wiring, and integration of individual GPCR signaling pathways at the cell surface and the nucleus. So his work has had a lot of impacts, and one of them is the studies that have advanced our understanding of the assembly of the GPC signaling complexes, the presence and function of such complexes on the nuclear membrane, and entirely new roles for G proteins as transcriptional regulators. And the second one is an innovation track, which has led to the development of a lot of new biosensors that capture the signalosomes of GPCRs in different contexts. So this has also seen the development of a second type of biosensor that tracks GPCR conformation, which is unique to his lab. And finally, there's a clinical translation to delay preterm labor and the testing of GPCR dimers as therapeutic targets in cardiovascular disease. And he has a new focus on GNB1 mutations, iPSCs, and in vivo of the lab's biosensors that has opened productive new vistas for his lab in cardiovascular disease, cancer, neurodevelopmental disorders, and also Parkinson's disease. So, Dr. Hébert, thank you very much. It's all yours.
Thank you very much for the kind introduction. I'm going to try to follow Kayla, give you a bit of a sense of what we're doing. And so I want to bring you from the single cell to the living brain. So, understanding disease evolution or understanding how signaling pathways are altered in disease or might be therapeutic targets in disease using our biosensor approaches, but moving away from sort of these heterologous expression systems into more disease relevant and patient relevant models. So we'll talk about that. For me, problem one is that we still miss a critical piece of the puzzle when we're thinking about disease, right? I use this slide in every single course I teach. I just can't stop obsessing over it. So I figure you can share my obsession as well. When someone gets sick and goes to see a physician, the disease is usually not, just starting then. It's ticked away in time. There's some underlying risk factors that could have been addressed, or there's some preclinical progression that could have been addressed. There are earlier events, but by the time the patient presents to the clinic, they're usually at the stage where there's not much can be done, in terms of disease modification, and we're focused on dealing with symptoms, right? So it would be cool to have systems where we could monitor some of this preclinical progression. The second problem for me as a GPCR person is that I've been thinking about how drug discovery has changed over the years and how it needs to continue to evolve if we want to make it meaningful to individual patients, if we're going to go down the road of personalized medicine. Historically, drug discovery was slow, but never left the realm of the biologically relevant. We used animal models, we used bioassays that were based on tissues, and we slowly went through the process of getting hits and then optimizing chemically leads. And the preclinical and the clinical studies were pretty successful, and we have a fairly high success rate if we go back in time to the historical era of drug discovery. And by that I mean the time spent doing drug discovery before we cloned everything. Since we cloned everything, we thought it might be simpler and more efficient to kind of just express each target, each gene that codes for a protein, in some heterologous expression system, and then develop high throughput screens where we could, look at 100,000 compounds a day or whatever in a simple, high throughput assay format. And it was beautiful until, it wasn't beautiful. And we lost the ability to kind of put new drugs in the pipeline. I mean, the failure rate became quite high. And so I guess what has happened in the intervening years for me is that I think there's some effort to restore the biology or the biological relevance to drug discovery by putting the relevant cell systems or the relevant tissues back into all these systems that we've developed to do high throughput screening. So not to throw out the baby with the bathwater, but to use more relevant models with all of the biosensor approaches that Kayla talked about, and then I'll talk about too. And ideally that will combine these two things where we have a high throughput, a reasonable debit rate for screening, but we never ignore the biology again. So I'll talk about two diseases today. Kayla already introduced you to cardiomyopathies, and Dr. Travenkov's questions kind of led you to think that they're caused by many different things, and that's true, right? So there are different flavors of cardiomyopathies that overlap in terms of disease progression and therapeutic intervention, but there's many that are caused by mutations that lead to dilated cardiomyopathies or hypertrophic cardiomyopathies. There's some that are sort of called channelopathies that have electrical disturbances, and then they kind of overlap with the spectrum of congenital heart diseases and neuromuscular disorders as well, right? So there's a genetic sort of association and a connection between these three different flavors, if you like, of cardiomyopathy. And in normal, progression to heart failure, we also see these three phenotypes too, and so they're interrelated, and they actually may follow one another, or they may not, right? So there's a lot of things to learn about. So the causes of cardiomyopathy are quite diverse, right? The easiest ones to model the way we're doing it are the ones that are connected to a particular genetic mutation. So that's our first patient population that we want to kind of just to test our ability to model would be in ones that we have some ability to predict what's going on in an individual myocyte. So we're looking at the disease of muscle straight. There are some that are caused by infection that are very important now, and I think even COVID-19 is driving cardiomyopathy. Dr. Chervenkov pointed out this connection between systemic immune-mediated disease and the development of cardiomyopathies, and he also acknowledged and Kayla confirmed that these are not easy to study in the context of the single iPSC-derived cell model, and you'd have to build some sort of organoid model or some sort of cardioid model to kind of get at that. So there are many other causes. The main other cause that we're interested in these days is that is actually preserved when we look at cardiac myocytes, is that when you have cancer patients who are treated with anthracyclines like doxorubicin and other drugs, they often show cardiomyopathies too. And the problem for those patients is sometimes it happens during the chemotherapy, sometimes it happens within a year of the chemotherapy, and sometimes it happens 20 years after the chemotherapy is over. And some of the changes or the risks that those patients might have might have been predictable before they were ever treated with these drugs, and that's something we're after as well. So the causes of dilated cardiomyopathy just are quite diverse. So can we use induced pluripotent stem cell-based modeling to understand dilated cardiomyopathy? And I think it started for me, again, I have to say with teaching, because I was interested in teaching about how these models were being developed. Joe Wu kind of started a revolution after Shinjo Yamanaka's work, where he could show that he could take blood or fibroblasts from patients who had familial forms of a dilated cardiomyopathy, and he could make them into cardiac myocytes. And he showed that they had all kinds of altered functions, calcium ion, so calcium handling, contractility deficits, abnormal distribution of sarcomeric alpha actinin, so they were highly disorganized. And he also showed that in the dish, you could model therapies. And he showed that if you stress these cells with epinephrine, they would often die, but you could prevent that death by treating with a beta blocker, right? So it recapitulated what was seen in some larger clinical trials, and it kind of gave us confidence that maybe there are things we could do at a patient by patient level with respect to understanding these diseases. So in our own little world now, in collaboration with Renzo Ciceri and with Nadia Gianetti at the Glen, we've started to collect and stratify patients and make stem cells from those patients, and then make cardiac myocytes from those patients. And you can see that you get cardiac myocytes and they beat, right? So this is exciting. It's not where we want to end up, because adult cardiac myocytes don't necessarily beat, but it is a simple way to identify that you've made a cardiac myocyte, right? So we're pretty excited about that. I still get all emotional when I see these. To come back to Dr. Chervenkov's question there, this, I don't know if you can see it, but this is our patient population. So about a quarter of them now, there isn't a suspected cause or an understood cause. About 15% of them are suspect to have a viral cause. 10% have a familial cause that can probably be linked to a particular mutation. 8% are patients who have been treated with chemotherapy or radiation. And these two populations here are our initial focus, I think, as we go forward to show proof of concept. And you can see a lot of other different causes that have been identified, including tachyarrhythmias, valvular disease, and there's some patients who have hypertrophic phenotype before they had the dilated phenotype. So we've got right now, well, as of last Friday anyway, 154 patients who have donated blood, and the Renzo Ciceri's group is very efficiently generating cardiac myocyte or generating stem cells from these patients, and we are making them into cardiac myocytes. So Kayla showed you this, and I guess what I view it as, it's, for someone who cut his teeth in cellular signaling, and who's concerned about the validity of cells used to model disease and therapeutic interventions, iPSCs and what we do are sort of a match made in heaven, right? So the process of getting the iPSCs, we have that part of the pipeline built. The process of making different types of cells to do, disease in a dish models, that pipeline is starting to go. We have reliable cardiac myocyte generation and culture, but we haven't gone too far down these other aims yet. I think Dr. Chveikov again pointed out that it's not only the myocyte that we need to worry about, right? It's co-cultures with other cells that are populating the heart. And ideally, you want to eventually build these into some sort of 3D structures that really mimic features of the disease. But the real cool thing for me is that all of this can be done while doing what we do in my lab, right? So we're a GPCR lab. We analyze signaling pathways. We're slowly getting back into being able to do electrophysiology on these cells. We're able to assess, the genomics of it, or to make disease-causing mutations using CRISPR or other technologies. And the idea is that we can use them at any stage along this spectrum from 2D to 3D to model disease, in individual patients, with the idea of being able to say something that improves their clinical prognosis. So I don't know if I skipped a slide. Yes, I did. And so we have this big toolbox of biosensors now, right? So we can measure events that occur, and Kayla told you about some of them that are designed to measure either the activation of pKa or the activation of ERK. We can localize them to different parts of the cell. So we can put them in the cytosol, we can put them in the nucleus, we can localize them to the SR, we can localize them to the cell surface, we can localize them to the mitochondria. The kits that we use, the toolkit that we've built or acquired are usually either based on resonance energy transfer, so bioluminescence or fluorescence resonance energy transfer, or these intensiometric biosensors that just capture fluorescence in response to something like calcium binding. The toolkit is big. It was hard to decide where to start. And so what Kayla showed you is that we started initially with two types of biosensors that either measure ERK or pKa in the cytosol or the nucleus. And COVID was useful for us because while we were not doing experiments, we actually had time to think. And instead of doing this bulk FRET to understand like a population level response, during our lockdown period, we were able to kind of think about, develop and build a single cell pipeline that Kayla described to do high content imaging to look at FRET responses one cell at a time. And she also said that we are starting down the pathway of being able to do hypertrophy one cell at a time. The idea is there you don't get an average response, you get a stratified group of responders, from either non-responders to high responders. And she showed you some of that. And we can capture the heterogeneity in the populations. We can identify leaders, cells that respond, and figure out why they respond or don't respond, right? And we can actually look at kinetic differences in individual cells. I mean, that are lost when you look at, or masked when you look at bulk readouts. And Kayla also kind of talked about this, that, the essential conundrum in looking at signaling in the cardiovascular system is that the same small set of signaling receptors is responsible for driving physiological responses like increased beating rate or increases in size, for physiological hypertrophy, as the ones that determine whether or not cells undergo pathological hypertrophy, or as the ones that decide whether cardiac myocytes live, get bigger, or die, right? It's the same ones that control whether we have a compensated hypertrophic phenotype to a cardiomyopathic phenotype and ultimately heart failure. So the same sort of activation of neurohumoral signaling pathways. So the sympathetic nervous system, the renin-angiotensin-aldosterone system, it's a vicious cycle that, when the heart perceives, or when the blood vessels perceive, or baroreceptors perceive a reduction in circulating volume and pressure, there's this vicious cycle of neurohumoral activation. all bearing down on the same signaling receptors. So, alpha adrenergic receptors, endothelin receptors, and angiotensin receptors, and then the GS coupled ones like the beta adrenergic receptors. There are some that are coupled to GI, but we're not really focused on them right now. But we know that the ones that are supposedly just coupled to these two pathways are also likely coupled to GI. So that's something that our systems give us the scope to explore. So we started, as Kayla showed you, with a smallish panel of ligands that are more or less therapeutic targets going forward, right? So looking at different angiotensin ligands, different beta or alpha adrenergic ligands, and then endothelin signaling as well. And she explained what she did. I don't want to get too much into the weeds of this, but so the real power of our approach is that we can now take patients who are either male or female and compare the responses down to the single cell level. And I don't want you to get too much into the granularity of these, but just notice this in looking at two patients, one male and one female, we already see that the patterns in response to angiotensin receptor signaling are distinct between male and female, and the patterns of responses for either alpha adrenergic or endothelinergic signaling pathways are distinct between male and female. There are nuances there that can be captured by our systems, and you probably know better than me that heart disease in women is a distinct thing from heart disease in men. And can we approach some of that those differences by understanding how the wiring changes differently, or how the wiring can be modified differently going forward? And I think that we now have the tools to do that using iPSC-derived systems from individual patients. And so to add a bit of flavoring to this, we also want to be able to kind of track morphological differences. So combining our signaling profiling, we're also trying to just stain the cells in different ways that allow us to track morphological classifiers so we can use to cluster patients. So the idea is to combine these morphological features with our signaling profiles to get at something like this, right? So starting with a cohort of 100 patients and control subjects, you want to be able to make a prediction about how each patient would respond to different drugs as they're given, right? So Kayla talked about that too. The issue that we face in doing that is that we can get to all of this, the signaling profiling, the cell painting, but what we want to do as a way station before we do that is, instead of using the cardiac myocytes, as they come out of the stem cell derived systems, we want to enhance their maturity. So I have a student who's working on that pretty hard to kind of figure out how to make these behave more like and look more like adult cardiac myocytes before we go into the clustering into disease subtypes. So there are challenges in doing that, and I just want you to be aware of that. But I think we stand a good possibility of being able to do a couple of things. Can we model future risks? So we're going to start pretty seriously with patients who have been treated with doxorubicin for breast cancer or for other things, right? 8 to 10% of women will get some version of doxorubicin-induced cardiac myocytes who are treated for breast cancer. 40%, 20% develop heart failure over 5 years of that patient population, and 40% if they're treated with dox and Herceptin, right? So, or if they're older patients. So can we model that risk? And Joe Wu has already kind of started that pathway, and we think we have some other tools that can allow us to kind of predict how sensitive patient cardiac myocytes might be to doxorubicin-induced injury, how we could test different anthracyclines that may be different in respect to their toxicities. We have a platform now to do that. And the other thing that I'm very interested in is something that John McMurray pointed out to me a couple of years ago, which that every patient who's treated for heart disease gets drugs pretty much in the same order, right? And it takes 6 months to up-titrate each of those drugs. And by the time you get to the one that actually works, that's a lot of time for disease progression. Why can't we sequence those things a bit differently than they're published in the trials, right? Why can't we ignore the guidelines, or at least not be held hostage by the guidelines? And we think that, using stem cell derived cardiac myocytes might be a way to more quickly test whether or not the sequencing in a particular patient might be effective in that patient. So those are the two things where I think we can use our system to impact therapeutic decisions. So are single cell-based models sufficient for real disease modeling? Well, I'm going to say no, because it's not true. I mean, there are things we can learn from individual patient-derived cardiac myocytes, but ideally you want to start building those into 3D kind of organoid structures that recapitulate the organization of the tissue that they come from. And I think that will give you better insights into disease modeling. That will give you a better bed in which to test therapeutic strategies. And ultimately, you know, if you think about what patients want, if they're going to have a heart transplant, well, I want you to grow the heart from my own cells. You know, I mean, we're not anywhere near that, but I think that's something that we're starting to talk about as a serious commitment now, right? And so, you know, the difficulty is that, you know, it's so easy to use the 2D systems and ignore all of the relevant things like, you know, do these tissues need to be vascularized? Do they need to have some sort of contact with the extracellular matrix? Do they need electrical and mechanical stimulation to drive all of that? Are there cell-cell interactions that are important? The answer to that is yes, right? So ease of use here and biological relevance here. So somewhere in the middle is where most of us end up. So is precision medicine, you know, going to be more than just weasel words that we put into our grants? And I think the evidence is that, yeah, but so we can already reprogram somatic cells, which is an amazing technology, really, into cardiovascular cell types, all of the ones that are relevant. You could even imagine putting in immune cells into that mix and looking at how the immune system interacts with that. That's beyond my pay grade right now. But you do recapitulate certain patient-specific and many disease-specific phenotypes that can be used to, if not individualize, at least stratify treatment strategies. And it allows you to model different diseases, right, to different extents. Yeah, where the rubber will hit the road is that we need to be able to do that in a sort of more scaled up way, and we need to do that in a three-dimensional way. And I think that that's where sort of platforms for drug discovery like ours and platforms for cardiotoxicity testing for things like stratifying patients for DOX or anthracycline treatment are now being incorporated into academic drug discovery pipelines, but more importantly, I think for drug discovery for real into pharmaceutical drug discovery pipelines. All right, so the last part is the third problem that I think about a lot is that it's still in vitro, even if it's patient specific. So what's the relationship between what we can measure in vitro and what's going on in vivo, right? So we've built all of these different platforms of biosensors based on either FRET or BRET, and we can test them in primary cells, we can test them in all kinds of cells. We can learn a lot about the signaling pathways that are coupled to an individual receptor. But in vivo is not in vitro. So we've been slowly building tools to kind of approach that problem more directly. So the idea is that not only can we understand signaling events downstream of a given receptor, but we can start to stratify ligands. We can start to look at different pathways that are sort of horizontal, that are coupled directly downstream of a receptor. And we can start to look vertically into individual signaling pathways to look at pathways downstream of the primary signaling pathway, right? We can capture all of these things very easily in vitro. But what do they mean when we go in vivo? So if we're going to start to do direct comparisons to the type of profiling that we can do in vitro, we need to start putting our biosensors into an in vivo context, right? So for us, obviously, you can't do this in the heart because you can't wire up some animal to have an instrument in its heart. But you can do that in the brain. So what I'm going to tell you about in the last few slides is just like moving some of our biosensor platforms from iPSC-derived cardiac myocytes into primary neurons, both cortical and in this case, striatal neurons, right? So we can express these biosensors. This is now the type of biosensor we're going to talk about, the same two that Kayla talked about, looking at the activation of either PKA or ERK in the nucleus or in the cytosol of primary neurons and in the living rat brain, okay? So the idea is that you can take your biosensors and you can inject them after making them into viral versions into the rat brain. So right here we're injecting sorry, I should go back and show that we can get nice responses for these sensors in primary neurons. So they work in primary neurons. And then you can go ahead and, oops, I skipped the slide. Yeah, and you could go ahead and imagine now putting them into a rat brain. And so you can do this with a confocal microscope, which you can attach to a rat's head, which will cost you about $400,000. Or you can do what we did, which was we had a purpose-built instrument that would just do fiber photometry and use a fiber optic cable that we attach to a rat's brain with an electrode array that is in the striatum. So we can show that the biosensors work first, as I said, in the primary neurons, and we're stimulating here the dopamine D1 receptor, and you can see activation of PKA. So when this sensor is activated, this site gets phosphorylated by PKA. It can now bind to this site on the other side, and it drags the donor and the acceptor closer together, and so you get an increase in FRET, right? So we can see that in primary neurons. Then what we would do is now we could package it up into cell type specific promoters, put them into recombinant adeno-associated viruses, and inject those viruses into the striatum, and then instrument the animals at the same time, right? And so a couple of weeks later, you can now make measurements in those animals. And not to get too much into the weeds again, but we're just going to talk about looking at either PKA or ERK as readouts in the cytosol, so in the cytosol or in the nucleus. And we know that both of those kinases act in the nucleus and in the cytosol. So we're interested in what's going on in those two compartments. So you can express the biosensor, as I said, and then now you can instrument the animal and you can inject dopamine or a dopaminergic agonist into the animal, and you can see that a dose-dependent response. You can see there's no effect of saline, and you can see that you can block it completely with an antagonist for the D1 receptor, right? So this is an in vivo experiment done in a behaving rat and living rat, right, which shows you that you can do dose response curves and you can see agonist and antagonist effects both. So we thought we would kind of expand the utility of our biosensors by taking it into a model of Parkinson's disease, so 6-hydroxydopamine induced destruction of dopaminergic neurons. And then we would monitor sort of what happens when these animals are treated with L-DOPA as therapy. It does deal with the motor symptoms, but eventually you get this dyskinesia, right? So Parkinson's is caused by degeneration of midbrain dopaminergic neurons, as you know, and so the deficit in striatal dopamine, we correct that with either dopamine agonists or with L-DOPA. But long-term treatment leads to reduced therapeutic efficacy and the progressive development of a lot of adverse consequences, including this L-DOPA-induced dyskinesia, which is actually, once it starts, you can't ever really have it regress back to normal. So we could see that when we make a unilateral lesion with 6-hydroxydopamine, you can see that if you look at forelimb use in the intact rat, it uses both forelimbs equally. But when you lesion the animal, you see it now uses only one limb as opposed to the other. And you can see that in the control animals, at a small dose of the dopamine ligand, you don't get much effect, but that is now sensitized when you lesion the animal. So these animals, because they're deprived of dopamine, their dopaminergic systems become sensitive or more sensitive to low doses of an agonist. And here you can see that this is a striatum imaged. The biosensor. Here's the intact side of the striatum labeled for tyrosine hydroxylase, and here's the lesion side where you've lost all of the dopaminergic neurons completely. So I don't know if I skipped the slide again. Yes, I did. Sorry. You can actually think about now modeling the different stages of the disease, right? You can model what happens when in the progression of the disease as dopaminergic neurons die, and you get this super sensitive state that I showed you. And you can also model what happens during the therapeutic intervention where you're giving L-DOPA and you want to see what happens to signaling events that drive or might be responsible for at least some parts of the L-DOPA-induced dyskinesia, right? So we want to understand how these events are changing in the living brain with respect to our biosensors. So our model was pretty straightforward. We would put the probes in and inject our either PKA or ERK biosensors after making the lesion. And then we would just test whether or not the biosensors expressed and evaluate if they had been sensitized. I showed you that in the previous slide. And then we'd give a therapeutic course with L-DOPA. We prime them for 2 weeks and then look at what happened both with respect to the development of the dyskinesia and with respect to what's happening to the signaling events, right? You can do that at the same time. Whoops. So in those rats, you get hypersensitization of both PKA and ERK responses, right? That's what you expect. And you also see that when you're measuring PKA, and so here's saline, again, no effect. PKA, you're getting a nice signaling response. And this is when you give DOPA, L-DOPA. And this is after that 2-week priming period. And you can see that these rats are already starting to show dyskinesias, right? And so this red line here shows you the extent of dyskinesia, and it exactly parallels the activation of the dopamine PKA signaling. And you see the same sort of thing when you look at ERK activation as well. So those signaling pathways are turned on when LID develops, and that hypersensitization of those signaling pathways is likely to be somehow involved in the rewiring of the rat's brain that leads to L-DOPA-induced dyskinesias. Whoops, I'll show you that rat again. So what we see in summary is that we can capture this supersensitive state as we get dopamine depletion. We can capture some of the events that occur in the development of L-DOPA-induced dyskinesia, and we can see which pathways might be both involved in that process, but also be actionable in that process if we want to think about therapeutic interventions. All of this is done in behaving animals, and all of these measurements are made in living rat brains. So we've developed techniques for recording FRET-based biosensors in vivo, and we can capture a lot of GPCR signaling events. We could detect the sensitization of PKA and ERK that happens when striatal dopamine is depleted in a model of Parkinson's disease. And we could track, and we could see, I didn't show you the data. Well, I guess I did show you the data, is that comparing this to this, you can see that they're still up, but they're not, they're a bit desensitized, right? So even though they are desensitized, what remains is still very well correlated to what's happening in terms of the animals being dyskinetic when they're being treated with, when they're being exposed to L-DOPA after they've developed the disease. So D1R signaling is primed to induce dyskinesia. And now we're setting out to kind of understand the different pathways that might be involved in preventing the development of dyskinesia or preventing the loss of or the hypersensitization of some of these pathways. So the questions that remain are, okay, we see this priming step. So as we go from sort of the priming when you have, before you have the dyskinesia to after you have the dyskinesia, is it there are fewer responding cells, or is it the response in an individual cell is reduced, right? So then we went back to the single cell high content imaging approach in primary neurons, where you could start to stratify these events. Whoops, why does it do that? You can start to stratify these events into individual neurons that are either low responders, medium responders, or high responders. And you could ask that question, right? So here we're now looking at a nuclear localized PKA sensor. And just to remind me to tell you that lots of stuff happens in the nucleus. So putting it there makes sense. And so we have this is now the study is done using the nuclear localized kinase. And you can see that you can now stratify both PKA, so AKAR, or ERK in the cytosol or the nucleus, and you can see that there are if you look at individual cells now, you can start to stratify the responses. And if you do that in a way that's more visually striking, you can actually do what Kayla did, where you look at each individual cell over time, and you can quantify how many are responding to the dopamine ligand, to an NMDA ligand, to the combination of those, or to direct stimulation of the pathway downstream, which is forceful, right? So you can see that if you look at all cells now, the response is reduced. But now if you just look at the responders, they're more or less the same, right? So it's not the individual response per cell, it's the total number of responding cells, which is changing. So we can, by doing this type of single cell analysis, we can ask questions like that about what's going on. Is it a population thing, or are some cells still able to respond and others not? And why is that? So just to conclude, I've been telling you about an imaging and analysis pipeline for large-scale single-cell imaging of both neuronal and cardiovascular cell cultures that express our fluorescent and FRET-based biosensors. We think it gives us a unique vantage point from which to observe signaling cell signaling dynamics, but across individual cells, into populations. But none of it makes any sense unless you have some sort of in vivo, correlation or in vivo confirmation of what you see in vitro rather. So to thank the people who did the work, well, you've already met Kayla. The other people who are doing cardiomyopathy modeling are Grace, Human, Kara, Allison, Darlene, and Karima. And then Jace and Hanan are the ones who did the in vivo imaging in rat brain. And Jace is also the one who kind of interfaced to help us build the single cell pipeline, both for his own work and for the work that Kayla showed you. And so with that, I also want to thank the people I collaborate with, Jason Tanny, long-standing collaboration, more recent collaboration with Nadia Giannetti and her team, and Renzo Ciceri and his team, Ida Kashif and Jeremy, and Nat, who helped us build our patient pipeline and the pipeline to get stem cells from those patients. And then long-term collaborations with Bruce Allen and Paul Clark here at McGill, David Chatanet, who's our ligand maker at the ENRS, and Tom Durkin, who helped me become a stem cell guy. And then the funders, CIHR, Heart Stroke, and a very generous private donation from the Courtois Foundation. Thank you. I'm happy to now answer or to discuss, and thanks again for inviting me. And I'll stop sharing my slides.
So thank you very much, Dr. Hébert, for that really exciting presentation. So any questions, please feel free to unmute. So just interesting that you can model future risks. So is the idea then with your studies, is the idea to cluster patients?
Yeah, I think the idea would be to, I guess, if we have for a large enough population of control subjects, we could start to ask questions about, are there generalities we can make about people's risks with being treated with doxorubicin? Are they sensitive out of before they're ever treated with doxorubicin, right? Or would they be less sensitive in terms of cardiomyopathy to another anthracycline or a doxorubicin derivative that would be different, right? So I think that we're going to test it first off in our patient population of women who have gotten it from breast cancer treatment, and Ida as I don't know if she's delivered them, but she just put she just told me that 7 iPSC lines from breast cancer patients are coming our way tomorrow. So we're going to start with that. But I think if we're going to build a sort of a broader risk stratification, maybe we start with not with patients, but with people in general, and see why some rather than others might be sensitive to different chemotherapies before they're treated with those chemotherapies. Like anyone who gets a cardiomyopathy, any therapy that leads to cardiomyopathy, we could test people for risks a priori.
Dr. Shevchenko?
Oh, hi. A really, really illuminating talk, and I really enjoyed it. And I was talking to Renzo about him 3 weeks ago because, I'm a transplant surgeon, so immunology is something I do quite a bit of and study. I'm not saying that I'm an absolute expert, but one of the things I've come across is in myocarditis and dilated cardiomyopathy is being implicated. The immune system has been implicated. And there's quite a few animal models where if you block interleukin 17 and the pathway of that cytokine, particularly, and it's relevant also to renal disease. A lot of our type 2 diabetic patients get into kidney failure. And again, this cytokine is very much implicated. This cytokine is implicated in NASH cirrhosis and fibrosis, and even in cancer. So my question to you, is there a potentially a theme that kind of like looks at this reperfusion injury pathways, which then may implicate some of the cascade of these Th1, Th17 cells and Tregs that then may modulate a lot of the diseases that we see eventually in some of these patients that kind of maybe target other organs, but that are very similar in the way they work. And one of the things I was wondering, the doxorubicin story and cardiomyopathy, is this involves some kind of an immune process as the cancer is being killed. The heart is some kind of an innocent bystander, very similar to some of the other autoimmune diseases that we see. Anyways, it's just my question. I would love to collaborate with you in the future.
Well, I'm going to say that your question was actually multiple questions, so I'll take them in the order that I remember them. The first was, can we use it to model some of the sort of reperfusion injury? I think a lot of that is still modeled best in large animal models for sure. Many of my cardiovascular colleagues who do it in dogs or in pigs. Could it be modeled in 3D organoid models? By the thing I didn't talk about is that there are people who are trying to build like a sort of patient microfluidic connected organ systems rather that communicate with one another, right? So I think that kind of approach is where you're asking if we could go, right? If you could kind of build the connections between organs into some sort of platform where you could test some of these things. The answer is yes, but the answer for me is not any time remotely soon, because we're not a microfluidics lab. We're not even yet a 3D lab. But I think there's a lot of things we could end up doing, once these platforms are a bit more available, a bit more better understood as to capacity and as to ability to kind of put them in place locally. But there are some. I really think that's the where we're going, right? Like to really personalize medicine. I tell students every time I teach them about this is that somebody should start the freezer company, right? Because everyone's going to want to start growing cells from their children or from themselves that they can use for things later. And that might include the mini-me, right? The organoid-based microfluidics connected systems. So
have you tested in your system the serum or the plasma of these patients to see how your system reacts to some of that, or no?
No, like it's still early days for us, right? So you're asking questions that seem like straightforward, like why didn't you try this or why didn't you try that? Because it's only been in about 2 years that we've been able to make muscle cells at all, right? So we're still kind of, how far can we push maturation? When is enough to actually start asking questions like you're asking, right? So at some point, we have to kind of agree that this is as good as it's going to get with this particular experimental system, and then just do it. Just start asking questions, and some answers will be right, and some answers won't be right. At the same time, we have to start pushing into 3D. I acknowledge that as a weakness too. So, no, we haven't.
It's great. It's great. Yeah, it's very good. Anyways, it's exciting what you're doing. Yeah.
Thank you.
So there's a question in the chat from Amani Hassan. And the question is cardiomyopathy associated with a specific type of cancer, or is it general?
Yes, and I see the second part of the question. Yeah. Why are we focusing on breast cancer? So there are, doxorubicin toxicity was not initially identified as in breast cancer patients, but it was identified in children who were being treated for pediatric cancers, right? So, and part of the solution for those patients has been, like, we do have some drugs that will prevent some of the damage induced by doxorubicin, iron chelators like desoxyrisane, and maybe some antioxidants like N-acetylcysteine. But it's become clear that breast cancer patients, so sorry, to finish that part, is that those kids do better on with it, and they don't get cardiomyopathies nearly as often when they're treated with desoxyrisane. And it was almost predictable that they might get cardiomyopathy if they didn't do anything. But there's no clear guides for breast cancer patients, right? We don't even really know if someone who's treated with doxorubicin is going to get it. So that kind of uncertainty kind of makes doctors use lower doses or be more careful and less aggressive in treating the cancer. So the focus on breast cancer, to answer the second part of your question, is because there is a defined patient population who gets dilated cardiomyopathies that we already have in our pipeline. So whether other types of cancers might be included in that, since we're only focusing on adults as subjects, we won't get the pediatric cancers. But it should be done. I just need about $8 more million a year.
So did I hear correctly from your talk that one of the aims is to be able to impact therapeutic decisions?
Mm-hmm. You know, that's a dream, but I realized that, you know, being stuck with the guidelines is something that clinicians cannot necessarily escape from easily. But a guy like me, I'm not stuck by the guidelines if I'm doing disease modeling in a dish. I can test things. I can try different orders in which drugs are given. I can try different combinations, right? And I think that what Packer and McMurray were proposing is that they were going to do that in patients. So I think that the two things work together, which is why we've kind of included him in our thinking going forward, right? I think that some clinician who's willing to kind of challenge the guidelines in that way is an ally to what we want to do.
That's wonderful. So any other questions? Please feel free to just unmute. Mike, do you have any questions?
No, it was a wonderful talk. Actually, I've seen most of his talk, but I did really like the FRET sensors, and this is something that I've always been interested in. So you managed to get them in the neurons, right, in a living animal, in the brain. Mm-hmm. In those slices, did you do brain slices as well, or was this simply imaging directly through the cranium?
So we didn't actually make like we thought we were going to have to do a halfway measure of showing that our biosensors worked in neurons and then worked in slices, because we didn't think it would work in brains right out of the box, but it did. So we didn't really go back down to the slices. It would be really cool to do that, because that's kind of like an organoid approach where you could start to look at different biosensors that you could put into different neurons and look for circuit changes in a more controlled setting. So again, my lab's not set up to do that, Mike, but that is something that we definitely thought about, that to explore the full power of the approach, you know, the 3D, preserving some of the 3D circuitry and 3D architecture. Slices make a lot of sense for neuronal preps to do that.
We're interested in looking at signaling as well, but I'm going to have to get in contact with you later on about that. We were interested in DRGs, so more on the dorsal horn. Yeah, so it would be amazing if we could figure out something together with these sensors.
Well, you know how to find me, so.
Yeah, but thank you, that was wonderful.
Pleasure.
Any other questions? Okay, if there are no more questions, I would like to thank Dr. Hébert for that wonderful talk, and Kayla, really exciting, and Rashad for the wonderful moderating. And thank you very much. It's been wonderful to hear your work. And I would like to thank everyone for coming to the seminar. Thank you.
Thank you all for being such a great audience.
Thank you.
Thank you. Take care.
Bye.
Bye-bye.